Hi everyone, welcome to all. This is Vidyam Academy's most expected question series for medical NEET examination. Today's topic is 12th Biology chapter number 5, Molecular Basis of Inheritance. And into this subtopic is the DNA, the search for genetic material. And now it's being presented by Rajiv Patel. Once again, let's welcome to all. Let's get started. The first question is, even Chargaff analyzed the base composition of DNA from various organisms and derived two rules. Which of the following correctly states Chargaff's rule? Option A. Amount of A is equal to C and amount of G is equal to T in any DNA sample. Option B. Amount of A is equal to T and amount of G is equal to C and A plus G is equal to T plus C. Purines equal pyrimidines. Option C. Amount of A is equal to G and amount of T is equal to C. Purines are equal and pyrimidines are equal separately. Option D. DNA base composition is constant and identical in all organisms regardless of species. So what is the right answer based on Irwin Chargaff analyzation? Yes, the right answer is option B. Let's see the detailed explanation why B is the right answer here. Chargaff's rules state two quantitative regularities in DNA base composition. First, the amount of adenine always equals the amount of thymine, A is equal to T. And the amount of guanine always equals the amount of cytosine, G is equal to C. This is the base pairing rule reflecting complementary hydrogen bonding between strands. Secondly, the total amount of purines A plus G always equals the total amount of pyrimidines, T plus C. This reflects the anti-parallel double standard structure where each purine one strand pairs with a pyrimidine on the other. However, the ratio A plus T, G plus C varies between species, making this ratio a characteristic of each organism's DNA. These rules were crucial evidence used by Watson and Crick when building the double helix model. That's what option B is the right answer for this question and rightly explained here. The next question number 2. Which of the following correctly describes the structural features of the DNA double helix as proposed by Watson and Crick in 1953? Option A. DNA is a single stranded helix with the bases on the outside and the sugar phosphate backbone on the inside. Option B. DNA is an anti-parallel double helix, two strands run in opposite direction, 5 to 3, 3 to 5 with bases on the inside held by hydrogen bonds and sugar phosphate backbones on the outside. Option C. DNA is a parallel double helix with the both strands running 5 to 3 in the same direction. Option D. DNA is a triple helix where 3 strands wrap around each other with each base pairing with 2 others. So what is the right answer for this question? Yes, the correct answer for this question is option B. Why and how? Let's see the detailed explanation. The Watson and Crick model, which is very famous. So double helix has several defining features. The 2 polynucleotide strands are anti-parallel. One runs 5 to 3, another runs 3 to 5 in the opposite direction. The sugar phosphate backbone, so deoxyribose sugar phosphate, forms the outer framework of the helix while the nitrogenous bases project inward and pair with each other through hydrogen bonds. A pairs with the T via 2 hydrogen bonds and G pairs with C via 3 hydrogen bonds. The helix makes one complete turn every 3 to 3.4 nm and contains 10 base pairs per turn. So each base pair is spaced 0 to 0.34 nm apart. The diameter of the helix is 2 nm. This right-handed B-form double helix is the form predominantly found under physiological condition in cells. That's what option B is the right answer. Question number 3. In Griffith's experiments with Streptococcus pneumoniae, what was the key observation that suggested the existence of transforming principle? Option A. Heat killed smooth S bacteria alone caused pneumonia in mice. Option B. Live rough or bacteria alone caused pneumonia in mice. Option C. Mice injected with a mixture of heat killed 5 bacteria and live or bacteria died of pneumonia, and live 5 bacteria were recovered from the dead mice. Option D. Mice survived when injected with live S bacteria because their immune system destroyed the bacteria. What is the right answer for this question? The right answer is option C. Griffith worked with 2 strains of Streptococcus pneumonia. The virulent smooth S strain with polysaccharide capsule with the causes of pneumonia, and the non-virulent rough S R strain without a capsule. He showed that live S bacteria killed mice, heat killed S bacteria didn't kill mice, and live R bacteria did not kill mice, all as expected. The remarkable finding was the 4th experiment, a mixture of heat killed S bacteria and live R bacteria killed mice, and live S bacteria were recovered from the dead mice. This meant the non-virulent R bacteria had been converted into virulent S bacteria, a permanent heritable change. Something from the heat-killed S cells had entered and transformed the R cells. Griffith called this unknown agent the transforming principle, and it was later identified as the DNA by Avery, McLeod, and McCarthy. That's what option C rightly explained and is the right answer for the question number 3. Let's move to question number 4. Avery, McLeod, McCarthy 1944 identified the transforming principle from Griffith's experiment. Which experimental approach confirmed that DNA was the transforming substance? Option A. They treated the exact with RNase, RNA diation eliminated transformation, proving RNA was the transforming principle. Option B. They treated the cell-free exact of S bacteria with protease, RNase, and DNA separately. Only DNase treatment destroyed the transforming ability, identifying DNA as the principle. Option C. They showed that proteins from S bacteria could transform R bacteria into S bacteria when added to the culture. Option D. They used X-ray crystallography to visualize DNA entering R cells during transformation. What is the right answer for this question? S. B is the right answer. Let's see why and how. Avery, McLeod, McCarthy prepared a cell-free exact from heat-killed S bacteria that retained the ability to transform R bacteria into S. They then systematically destroyed each class of biological molecule in the exact and tested whether transformation still occurred. Treating the exact with the protease destroys proteins, transformation still occurred. Treating with RNase destroys RNA, transformation still occurred. Treating with the DNase destroys DNA, transformation was completely abolished and our bacteria remained non-virulent. This elegant elimination experiment conclusively identified DNA as the transforming principle, the heritable factor responsible for converting non-virulent or bacteria into virulent S bacteria. Despite this clear evidence, many scientists still believed proteins were the genetic material, and it took the Hershey's experiment with the pace to fully convince the scientific community. That's what option B is the right answer for this question. Let's move to question number 5. Hershey and Chase 1952 used bacteriophage T2 to confirm the DNA, not protein, is the genetic material. What radioactivity labels did they use and what did they result now? Option A. 32P labels protein, 35S labels DNA. DNA entered the host cell confirming protein as genetic material. Option B. 35S labels DNA, 32P labels protein. Protein entered the host cell confirming DNA as genetic material. Option C. 32P labels DNA, 35S labels protein. Only 32P DNA was found inside infected bacteria, while 35S protein renamed outside in the page goose. Final one D, both 32P and 35S labeled DNA, both were found inside infected bacteria, proving DNA is the genetic material. What is the correct answer for this question? The answer is option C. Let's see the detailed explanation. Hershey and Chase exploited the fact that DNA contains phosphorus but no sulfur, while protein contains sulfur but no phosphorus. They grew one batch of T2 pages in medium containing 32P radioactivity phosphorus to label the DNA, and another batch in 35S radioactivity sulfur to label the protein coat. When 32P labeled pages infected bacteria, most of the radioactivity was found inside the bacterial cells after centrifugation. DNA had entered, so when 35S labeled pages infected bacteria, almost all the radioactivity remained in the supernatant, the phase protein coats called ghosts. After centrifugation, protein had not entered the cells. The progeny phases produced carried 32P labeled DNA but not 35S protein, proving the DNA and not protein was injected into the host, carried the genetic information to produce new phases. That's what option C is the right answer. Question number 6. A DNA nucleotide is made of 3 components. Which of the following correctly identify all 3 components and the bonds that link nucleotides together in a polynucleotide chain? Option A, deoxyribose sugar plus nitrogenous base and phosphate group nucleotide linked by hydrogen bonds between bases. Option B, ribose sugar plus nitrogenous base plus phosphate group nucleotide linked by 3 to 5 phosphodiester bonds. Option C, deoxyribose sugar, nitrogenous base, phosphate group, nucleotide linked by 3 to 5 phosphodiester bonds. And final one D, deoxyribose sugar plus amino acid plus phosphate group nucleotide linked by peptide bonds. What is the right answer for this question? Yes. Let's see your guess whether right or wrong. Option C is the right answer here. Let's see the detailed explanation. Each DNA nucleotide consists of 3 parts. A pentose sugar, specifically 2-deoxyribose, which lacks the 2-OH group present in ribose. One of 4 nitrogenous bases, adenine, guanine, and cytosine and thymine. And a phosphate group. The base is attached to the 1 carbon of the deoxyribose and the phosphate is attached to the 5 carbon. Adjacent nucleotides in the same strand are conveniently linked by 3 to 5 phosphodiester bonds. The 3-OH of one nucleotide reacts with the 5-phosphate of the next, realizing water and forming the bond. These phosphodiester bonds form the covalent backbone of the single strand. The 2 complementary strands of DNA are held together not by covalent bonds but by hydrogen bonds between the complementary base pairs AT with 2 hydrogen bonds, GC with 3 hydrogen bonds. That's what option C is the right answer. Next, move to question number 7. Which of the following correctly distinguishes DNA from RNA in terms of their chemical structure and function? Option A, DNA has ribose sugar and uracil. RNA has deoxyribose sugar and thymine. Option B, DNA is the genetic material that stores formation. RNA has deoxyribose and is found only in the nucleus. Option C, DNA has deoxyribose sugar, thymine and is double strand and more stable. RNA has ribose sugar and uracil instead of thymine and is usually single stranded and less stable. Option D, DNA and RNA have identical sugar phosphate backbones but differ only in their base sequences. What is the right answer for this question? Could you guess the right answer? Yes, the option C is the right answer for this question. Let's see the detailed explanation here. DNA and RNA differ in 3 key structural ways. First, the sugar. DNA contains 2-deoxyribose, no OH at the 2 carbon, while RNA contains ribose, has OH at the 2 carbon. This makes RNA more reactive and less chemical stable than DNA. Second, the bases. DNA uses thymine, T, while RNA uses uracil, U in place of thymine. So both pair with adenine, but uracil lacks the methyl group the thymine has. Third, the stranding DNA is most always double strand. DNA is most always double stranded, providing structural stability and redundancy for accurate replication, while RNA is a typically single stranded but can fold into complex secondary structures. These structural differences directly relate to function. DNA's great stability makes it deals as the long-term store of genetic information, while RNA's versatility, mRNA carries messages, tRNA carries amino acids, rRNA forms ribosomes, serves the dynamic process of gene expression. That's what option C is the right answer here. Next, move to question number 8. So here match the column 1, scientists and pay, with column 2 is the key contribution of them. The first one, Griffith in 1928. Second one, Avery, McLeod and McCarthy in 1944. 3, Watson and Crick in 1953. 4, Hershey and Chase 1952. So here are the options. Option 1 to A. Used 32P and 35S to show DNA, not protein, enters host cells during phase infection. Second one. B. Showed transformation and Streptococcus, a transforming principle, converts all bacteria to S. C. Used DNase to identify DNA as the transforming principle. D. Proposed the anti-parallel double helix model of DNA structure. So here are the options. A. 1B, 2C, 3D, 4A. B. 1C, 2B, 3A, 4D. C. 1B, 2A, 3D, 4C. D. 1D, 2B, 3C, 4A. What is the right answer? The correct option is A. 1B, 2C, 3D, 4A. Let's see the detailed explanation. So Griffith 1 matches B because his 1928 experiment with the Streptococcus pneumonia showed that a live or bacteria could be permanently transformed into S bacteria by something from heat-killed S bacteria, which he called the transforming principle. Avery, McLeod, McCarthy 2 matches C because their 1944 biochemical dissection used DNase to destroy the transforming activity, conclusively identifying DNA as the transforming principle. Watson and Crick 3 matched to D because in 1953, they proposed the anti-parallel right-handed double helix model of DNA structure using data from X-ray crystallography. So Rosalind Franklin and Maris Wickens, Chagav's base pairing rules and model building. Hershey and Chase is here the 4 which matches A. Their 1952 blender experiment with 32P labeled DNA and 35S labeled protein pages conclusively showed that only DNA entered the host cell during infection. That's what option A correctly and rightly matched here. Let's move to question number 9. We have assertion A, DNA is more suited than RNA to serve as the permanent genetic material in most living organisms. Reason R, the 2OH group present in the RNA mix is more reactive and the prone hydrolysis compared to DNA, which lacks this group, making DNA chemical so more stable and therefore a more reliable store of genetic information over time. So here are the options. Option A, both A and R are true and R is the correct explanation of A. B, both A and R are true and R is not the correct explanation of A. And C, A is true but R is false. And D, A is false because RNA is equally good as DNA for storing genetic information. What is the right answer? The right answer of this question is option A. Both A and R are true and R is the correct explanation of A. Let's see. DNA is the preferred genetic material in nearly all organisms with the exception of RNA viruses. Its greater chemical stability makes it suitable for long-term storage of hereditary information that must be faithfully replicated over millions of cell divisions. Reason R is also true because it gives the precise chemical basis. RNA contains ribose sugar with a free 2OH group adjacent to the 3 to 5 phosphodiester bond under alkaline condition or enzymatic action. This 2OH can act as a nucleophile and attack phosphodiester bond, leading to the self-hydrolysis and strand breakage. making RNA inherently unstable and short-lived. DNase deoxyribose lacks this 2OH group, eliminating this degradation pathway and conferring much greater stability. R directly and correctly explains A. Chemical stability from the missing to ZOH is the molecular reason. DNA is the preferred genetic material in nearly all organisms, with the exception of RNA viruses. Its greater chemical stability makes it suitable for long-term storage of hereditary information that must be faithfully replicated over millions of cell divisions. Reason R is also true and gives the precise chemical basis. RNA contains ribose sugar with a free 2OH group adjacent to the 3 to 5 phosphodiester bond under alkaline condition or enzymatic action. This 2OH can act as a nucleophile and the attack of phosphodiester bond, leading to self-hydrolysis and strand breakage, making RNA inherently unstable and short-lived. And DNase deoxyribose lacks this 2OH group, eliminating this degradation pathway and conferring much greater stability. R directly and correctly explains A. Chemical stability from the missing 2OH of the molecular reason DNA is the preferred genetic material, confirming option A. The final question of this topic is, arrange the following landmark experiment and discover in correct chronological order. So here P statement, Griffith's transformation experiment, transforming principle identified in a streptococcus. Q, Avery, McLeod and McCarthy biochemical identified DNA as the transforming principle. Or Chagaf derives base pairing rules A is equal to T, G is equal to C in DNA. Yes, Hershey and Chase confirm DNA enters host cell during phage infection using radioactive labels. T, Watson and Crick proposed the double helix model using X-ray crystallography data. And U, Rosalind Franklin obtains photo 51 X-ray diffraction images of DNA showing helical structure. Option A, PQRSUT. Option B, PQRUST. Option C, PRQSUT. Option D, PQRSUT. Guess the exact correct chronological order. PQRSTU. So what is the right answer? Option B, PQRUST is the right answer, because P 1928 and Q 1944, R 1950 and U 1952, yes 1952, again T is 1953. Let's see the detailed explanation. So Griffith's transformation experiment was 1928, the first evidence of inevitable transforming agent. Avery, McLeod, McCarthy in 1944 identified this agent as DNA using DNase elimination. Chaga published his base pairing rules in 1950. A is equal to T, G is equal to C, providing quantitative data crucial for model building. In 1952, Rosalind Franklin obtained the famous photo 51 X-ray diffraction image showing the helical nature and dimension of DNA. And the same here, Hershey and Chase confirmed DNA as a phase genetic material using 32P and 35S labelings. Finally, in 1953, Watson and Crick synthesis all available data, Franklin's X-ray data and Chagaf's rules and Wilkins' measurement into the complete anti-parallel double helix model T, publishing it in Nature. This 25-year sequence from Griffiths to Watson Crick is the most important experimental arc in molecular biology. So here we explained 10 most important and expected questions for your upcoming NEET examinations. And next, I will come up with another topic which are very important for your NEET examination. Till so far, I would like to conclude this session with very well goodness. Thank you, and thank you so much to all.